So now in this next video, which we'll entitle Sea Urchin Gastrulation SUG2, we're going to be looking at the specific sequence of events that are involved in the gastrulation of a sea urchin blastula, or embryo that's developing. These sequence of events are highlighted very nicely, and as we go through this, be sure to follow figure 47.8. So now, let's take a look at the events that occur during gastrulation, and hopefully, when we cover all of these events, we'll cover all of the characteristic highlights that we saw in the previous video. So, first and foremost, let's begin at the start of gastrulation. The start of gastrulation is going to occur at the vegetal pole. So, sea urchins are going to have these developing embryos that do have a bit of yolk in them, and there's going to be one side that has the vegetal pole, let's say. That's where the yolk is technically going to be near, and that vegetal pole will be with things called mesenchyme cells. This is a new term to remember, mesenchyme cells. So this vegetal pole with the mesenchyme cells, something's going to happen here, and specifically it's the first characteristic highlight. The cells at this part of the developing embryo are going to migrate. Cells migrate. Guess what? We've already covered the first characteristic highlight of sea urchin gastrulation, which was cell migration. Here it is. Cells migrate into a structure that's within the blastula called a blastocele. So now these cells that were originally on the outside of this blastula are now going to migrate into the cavity that's called the blastocele. When they're moving into this area, they're going to be doing this for a purpose. There's a specific reason. Some of these cells, some will later, as a result of developmental cues and signals, they will secrete something called calcium carbonate. CaCO3 for chemically short, let's say. So they secrete CaCO3. What is of use for this chemical, this compound? What's the purpose of it? Of course, if you remember, CaCO3 is a critical component of an internal skeleton of many organisms, and thus, later on, as a result of this cell migration process during gastrulation, eventually, you will form what is known as a simple internal skeleton that the sea urchin possesses upon complete embryonic development. And that's our first sort of characteristic highlight in a nutshell. What's the purpose of cell migration? The eventual purpose of cell migration in the gastrulation of a sea urchin is to form this internal skeleton as a result of these processes highlighted here. So there's our characteristic highlight done. Next characteristic highlight to look for, let's begin over here. Let's say there's also going to be some other events going on at that vegetal pole. So let's write this down. So let's go back to that vegetal pole. There's going to be other cells there at vegetal pole, comma. Other cells, not these mesenchyme cells, but other cells will do something different. They will slightly flatten. Okay, so this is the process they will undergo during gastrulation. They will slightly flatten. When they slightly flatten these other cells, they're going to cause this structure. This causes the whole vegetal pole area, VP for vegetal pole area, to buckle inwards. Keyword here right now is inwards. This would mean that you have essentially created at the vegetal pole an invagination because of this inward moment, an event that has occurred. And there it is, invagination, which was our second characteristic highlight, has just been done during gastrulation in this sequence of events. The invagination will happen directly into the embryo, as we said in the previous flowchart and video. So there we go, we've got two characteristic highlights done during sea urchin gastrulation. Let's move forward. Next, what we're going to do is form the archenteron. The endoderm cells will form the archenteron. And if you remember the archenteron, you can remind yourself on the side here, this is the future digestive tract. It's going to be the future digestive tract lumen specifically. So these endoderm cells will do this during gastrulation. Specifically, this is going to happen in a very sort of stepwise manner, much like everything else. What's going to happen here? We're going to have those mesenchyme cells. Again, they're going to show up again. Mesenchyme cells will show up again. Remember how we said some will later secrete? Well, some others are going to do something else. Mesenchyme cells that are located at the tip of the archenteron, produce a structure that's very important in digestion, or later in digestion, in the development of digestion, I should say, called the philopodia. So this is a term, new term to remember. Mesenchyme cells at the tip of the archenteron produce philopodia. What are philopodia, and what use are they during gastrulation? These are going to be very thin cell extensions. They are thin cell extensions that extend towards the blastocele wall, that extend, thus their extensions, toward the blastocele wall. So we still have this blastocele, this central cavity within this developing blastula that's trying to turn into a gastrula. So these thin extensions will extend towards there. Now, what they're going to do is eventually they'll coalesce and sort of combine and create this major contraction event. They eventually contract. When all of these philopodia, if you look at figure 47.8, make a one-time sort of cohesive contraction event, they're going to do something very purposeful. They're going to be taking this archenteron and dragging it. This contraction that's occurring is simply going to drag the archenteron, this interior lining right now, across the blastocele. And this is going to cause later some significant events of gastrulation, as we'll see. And we'll leave it at there for right now. So this is our sort of development of the archenteron and consequence of that development. This archenteron is being dragged across the blastocele. What's the purpose of that, and why are we doing that? Well, this is occurring because now what's going to happen is, as this dragging is occurring, the blastopore, which is the open end of the archenteron, the first open end, actually, as a result of the dragging, fuses with the blastocele wall of the overall structure. So it fuses with the blastocele wall, and the key here is fuses. Now, the blastopore, remember, is the open end of the archenteron. We can remind ourselves that over here. It's the open end, and this is essentially the anus of the organism. But what we have to do now is create that blastopore. And this site of fusion, when we have this fusion with the blastocele wall, as you'll see in figure 47.8, we can call this site of fusion, and it becomes this site of fusion, that development of the mouth opening. It causes the mouth opening to develop directly as a result of this fusion event that was thanks to the dragging of the archenteron across the blastocele. Notice how we're building upon everything that's happening prior and then subsequently. So now overall, as a result of this fusion, as a result of this development of the mouth opening, this creates a digestive, or completes, it creates a, I should say, complete on top of here. Squeeze that in. It creates a complete digestive tube, because that digestive tube, any tube that you know of, has two openings, and thus this is because it will be with both the mouth opening, which is the second opening, and also the first initial blastopore anus opening that we saw. Overall, as a result of this, we can finally sort of summarize on the side here the following. Overall, you're going to develop, as a result of all of these things happening simultaneously and after one another, essentially, you're going to get a gastrula. And the gastrula will be with 3 germ layers, as we saw and as we highlighted in the gastrulation flowchart. These 3 germ layers are actually going to be covered in something called cilia. So cilia are just finger-like extensions that are going to be very important later on in feeding. Okay, they're going to be important in feeding, something to just keep in mind, the cilia that is, and also movement of the sea urchin. So the sea urchin has this specific sort of overall event and ending that forms and allows for the feeding and movement eventually in its adult stage. And then finally, as a result, I want you to remember about the sea urchin gastrulation. One thing is that all of this development, all of this developing is happening externally. Now, why is it happening externally in sea urchins? That's because they fertilize their egg, sperm and egg, externally, outside of the parent. So this is not happening within a womb structure. It's not happening within the mother. This is all happening externally in the water. All of the events that we saw, and that covers our look at sea urchin gastrulation. Notice how we did cell migration, we did the invagination, the archenteron, the blastopore. We even formed the second opening, which was the mouth, and now we can move forward and look at a different gastrulation process within an organism known as the frog.